just a quick reminder, we have our student interns presenting today. So our presenters today are going to have about 10 minutes for their presentations. That's why this is a little bit of a longer presentation. We've got 8 presenters for our webinar today. So for those of you who are joining from the consortium, there's a roll call sheet to sign in, and just make sure that you stay muted unless you're one of our presenters, and we'll be recording. So we'll put the recording up in the Google Drive as well as the slides. You can also check those out after the presentation. So to give folks a little bit of a background for today's webinar, we have presenters from our underrepresented student internships with us today. So in case you have been following along or need a little more background, our HuBMAP underrepresented student internship program, these are underrepresented as defined by NIH undergraduate students who got the opportunity this summer to work with members of HuBMAP labs all across the country in different labs. They were virtual experiences this summer, which was great because we got students from all over the country as well who got to participate in these. So thank you so much to our HuBMAP member labs who hosted students this summer. It was a really great opportunity, and we had 8 students participate, and this was the first time that we had this student internship program. So thank you so much to everyone who participated and helped make this program a success, and we're so excited to hear from our students today. So to kick us off, I will
Megan?
Yes.
I think Zorina and I wanted to say a few words, if that's okay.
Sure. Yeah, go ahead.
Hi, this is Ajay Pillai. I wanted to offer up a special thanks to all the students, the PIs in various labs who actually, in trying situations, decided to give them a chance and give this program and project a chance. And a lot of people generally, Megan and Robin and other folks at the IEC. Who else? There are a number of things that Zorina managed to corral a number of NIH folks to talk to the students throughout the 8 weeks that they were part of this program. I don't know if any of them are here today, but even if they're not, we need to thank them. And of course, Julie as well, who helped with a lot of interesting ideas and suggestions for making life for the students better. And I think I have seen a sort of a draft of these presentations, and they are fairly exciting to listen to. And it seems like, even though the virtual setting was not the most conducive of all settings to learn a new field and a new domain, the students seem to have, along with their PIs, managed to do a great job, as you will see. We hope to continue this for the next coming year, and Zorina and I will be reaching out to the PIs so that we get a broader range of volunteers, and hopefully, the stars and everything else will align next year so that we'll actually have a in-person opportunity. And thanks again to everyone. That's it. I'm done.
I just want to say thank you to everyone who was already involved, as you heard from Ajay, students first and foremost, and then the labs and PIs who hosted them, but also all of you for calling in today. And for us, it's a big graduation event, and you are here to support our students. I will ask you to please help them with your feedback, but understand that this was a very limited amount of time that they had at their disposal, and they actually did an amazing job given all the challenges we had in this virtual world. It usually works best if you already know the people, then you can go virtual, but think about the fact that they didn't know their labs. They didn't know each other. They didn't know us. So they really did an amazing job. And we are here to celebrate their learning experience. We also asked them all to give us feedback to know how to make better the next series of internships. Hopefully those will be in person, but everything we learn is going to be of great value. So I want to invite you all to support the students, ask questions, and encourage them. And that's that. Thank you.
Okay, thank you, Ajay and Zarina. So we can get started, and I will introduce our students and then we will jump into the presentation. So we've got Stephanie, who worked with the Gregory Lab at UPenn. We've got Tatiana, who also worked with the Gregory Lab. We've got Oge Chukwu, who worked with the O'Neill Lab at UPenn. We've got Kate, who worked with the Penn Image Computing and Science Lab. We've got Casey, who also worked with the Penn Image Computing and Science Lab. We've got Injil, who worked with the Nolan Lab over at Stanford. And then we've got FJ, who worked with the Kim Lab at UPenn, and Roselkis, who worked with the Gellenberg Lab at Harvard Medical School. So 8 awesome students joining us today from across a lot of different universities, working with different HuBMAP consortium members. So we're excited. Stephanie, if you would like to start us off, I will stop sharing my screen and you can show us your presentation.
Sure, thank you. Okay, so hello everyone. My name is Stephanie Bobadilla Regalado, and I was an intern of the Gregory Lab at the University of Pennsylvania this summer. Today, I will be talking to you about some of the unique upregulated genes in the female reproductive tract, specifically the immunoglobulin genes. So let me tell you a little bit more about our project. Our ultimate goal was to learn what cell types are present in the 4 main organs of the female reproductive system. And once we know that, then we can spatially map out where these cells are located within the tissue. So let's talk about how we are conducting the investigation. The data that I'm going to present today comes from somebody who is undergoing gender redefinition treatment, and they were going through high levels of testosterone. And so the O'Neill laboratory at the University of Pennsylvania did our tissue dissociation, and they cutted the cervix into 22 layers, and they performed single cell suspension, which is just a culture where the cells are allowed to multiply, so that we can obtain high resolutions of what cells are located in each splice. So then here comes the fun part, which is the single cell RNA sequencing. And this approach provides the expression profiles of individual cells. And so within the data, you can identify the different cell types. And according to the Cystic Fibrosis Center at Stanford, the human genome is estimated to contain approximately 80,000 to 100,000 genes. So you can imagine how much data we get from this method. But a common method that is used with this single cell sequencing is dimensionality reduction by Umap, which is uniform manifold approximation and projection. So this simply refers to the process of reducing the number of attributes in a data set. And so we do lose some of the variability from the original data set, but it helps reduce the noise. And at the end, there are more pros and cons with this method. And so I'll talk more about Umap in our next slide. But just to finish this one off, after we create the Umap plot, you can spatially map out where these cells are in relation to the organ's tissues by using FISH. And so FISH is a method for detecting particular RNA molecules within the cells via fluorescence microscopy. And basically, you just attach a fluorescent dye to a piece of DNA, and then you target the RNA sequence within the cell, and that's how you detect the location of individual RNA molecules. So that's the overall method section. So now I'll move on to the data. So like I mentioned before, all of our data After all of the data correction, at the end, you get a plot like this one, which is after UMAP. Each dot represents a single cell, and the dots or cells that are clustered together are more similar to each other. So like these ones over here, or these ones over here. And then you can identify lots of different cell types by looking at the genes within those clusters. And so for this presentation, I chose to focus on cluster 12, which is just this one right at the bottom here, isolated. And I wish I could go over each cluster and tell you the main genes found in them, but due to the short amount of presentation time, I will only be going over the top 5 upregulated genes in cluster 12 as seen in this table. And so the p-values for all of these are very significant. IgDH is a gene in cluster 12 and is upregulated 9.72 times, which makes it the most common gene found in that cluster. It has a p-value of 5.17 times 10 to the negative 17. And you can note that all the upregulated genes in cluster 12 are immunoglobulin genes, or otherwise known as antibodies. And so this G chain right there, that doesn't start with Ig, but it is the joining chain of multimeric of 2 antibodies. So it still falls into that category. And so there are 3 main antibodies, and yes, there are a total of 5, but most commonly there are 3, and those are IgA, IgG, and IgM. IgAs protect body surfaces that are exposed to the environment, so these are found mostly in the nose, ears, eyes, and other parts. IgGs are found in all body fluids, and it controls infection of body tissues. These are the ones that protect you against an infection by remembering what germs you have been exposed to in the past. And then there are IgMs, and these are the largest antibodies, and they are the first to respond to an infection. And so how do these antibodies have to do with the female reproductive system? Well, in a study done by Pregnancies et al., the levels of these 3 antibodies are dependent on the menstrual cycle and are influenced by hormones. And so this table you see here in this slide is done by Gonzalez et al., where they compare the immunoglobulin levels by sex. And so you can see that females have less IgAs, but they have more IgGs and IgMs in serum. And so now in seminal fluid, Pregnancies et al. Concluded that IgGs and IgAs are most prevalent in men. And so based on the published data and our data, we do expect to see these immune cells in the female reproductive tract, which is why we see them in cluster 12. So with a little bit more time, we can analyze if our donor is transitioning to a collection of male-like genes. And so what is next for our lab? Well, this was the data for only the cervix, so we still need to move on to the next 3 organs of the female reproductive tract and do exactly what we just did for this, but for those organs. And then we can spatially map this using the FISH method mentioned earlier so that you can detect the location of individual RNA molecules within the tissue and or the organ. And like I mentioned before, this donor was undergoing hormone treatment, so there can be a comparative analysis with no treatment and determine if there are different cells that dominate each cluster. And so none of this would have been possible without Dr. Gregory and Ron Guo from the University of Pennsylvania. Ron was the one who made the libraries. Thank you to the O'Neill Lab for taking the samples and conducting the tissue dissociation. And finally, I would like to thank the NIH and HuBMAP for funding this project and giving me the opportunity to be a part of this. And I'm happy to take any questions at this time.
Thank you, Stephanie. That was a great presentation. If anyone has any questions for Stephanie, feel free to put them in the chat. And you can also feel free to continue to drop those in the chat, Stephanie, if you want to just keep an eye on that too. You can always answer them in the chat. Sure. So if anyone has any questions for Stephanie, definitely drop those in there, and we will go on to presentation 2, which is Tatiana.
Hi, everyone. Great job, Stephanie. The presentation was amazing. Okay. As you guys may know, Stephanie and I were privileged to work in the same lab, so we may see some overlap in our presentations. And yeah, so my name is Tatiana Gonzalez. I've been interning with the Gregory Lab for the past 2 months, and I focused my research on the effects of hormonal therapy on the single cellular level. And before I go into that, I'd like to talk a bit about HuBMAP and our contributions. So HuBMAP, the overall goal is to basically study human tissues down to the single cellular level and to be able to create a 3-dimensional map of those cells. I find this really interesting because I think it will give a lot more insight on how cells work together, and as well as to understand where they are actually located spatially and the genes that are involved with each cell. Another thing I find really special about this project is that we're just such a diverse group of organisms. So we have to make sure we study both sexes. We need a variety of ages, a variety of ethnicities, because all of our bodies are different. And that leads me to what our lab was focused on, which is the female reproductive tract. As Stephanie mentioned, we'll be studying fallopian tubes, ovary, uterus, and the cervix. The way we study this is we have organ donors who will donate their organs to us, and some of the requirements that we have are that the organs come from someone who is premenopausal, and that all of the organs are from one donor, just to provide consistency throughout our research. Something interesting that happened, though, is one of our organ donors is actually undergoing a gender reassignment. So they're being hormonally treated through testosterone therapy. And I wanted to see what those effects had on the cervix. So basically, testosterone and hormonal therapy, the goal of this is to bring estrogen levels down and testosterone levels up. And I think it's important to talk about what average testosterone levels are for a female, male, and then female transitioning to male. So for a female, it's 15 to 70 nanograms per deciliter of blood. And for male, testosterone levels range from 280 to 1,100 nanograms per deciliter. And then from female to male, the testosterone goal is 400 to 700. So you can see it's a big jump from 15 to 70 and going to 400 to 700, which led me to my research question: how does this affect the cervix on the single cellular level? In order to conduct my research, I had to set up a few goals. First was to determine all the cell types present in the cervix. Next was to make comparative observations between cell types in a hormonally treated cervix versus a non-hormonally treated cervix. And finally, it's to research why certain genes are expressed more than others between the two, and what that means in terms of hormonal therapy. So here we have our methods. First, this is a very broad method list, just for the sake of time. But first, we have tissue dissociation, where you're slicing the organs into very small sections for higher resolution to see more in detail what cell types are there. Next, we have nuclear isolation, which is the actual breaking open of the cell in preparation for gene expression analysis using 10x genomics. And that's where I come in here at step 3. And for our final step, it's spatial transcriptomics using FISH. Our lab has not gotten to that step yet, but that is kind of our end goal. Okay, so here we have a Umap of what seems like a typical cervix. So each dot represents a single cell, and they're clustered together in terms of similarity to each other. And what I meant by a typical cervix is one that has not undergone testosterone treatment. And this one actually has undergone testosterone treatment, and it is this little cluster here in purple. We call it cluster 13. And what's special about this is that this cluster here is unique to cervix that has undergone testosterone treatment. So one that would not have this cluster here at all. So this is kind of like the focal point of my research. As you can see here, this cluster is isolated. Over to the right, we can see it's cluster 13, that's what we're calling it, and it only has 120 cell types in that cluster, which is the least amount in comparison to the rest of the clusters. What I really want to focus on is this area down here, and this is our gene expression. So the reason I included this picture is because for comparative purposes, if we see here at cluster 13, we can see how upregulated these 3 genes are. But when you go across and look at the other clusters, they're very downregulated in comparison. So we'll look at the first one, which is MIR31 or MIR31. In cluster 13, this gene is upregulated 9.38 times. And given the p-value, it is highly significant. So when you look over here, these are all in the negatives, meaning they are very downregulated and not being expressed in these cell types. So I found it very interesting that the genes being expressed in cluster 13 are very, very different from genes being expressed in the rest of the clusters of the cervix. And I will go over 3 of them for the sake of time. Okay, so what gene expression tells us about effects of hormonal treatment in the cervix? So first, we are starting off with MIR31. This was the most upregulated gene, and its expression is often linked to cervical carcinoma as well as colorectal cancer. And it is also linked to Duchenne muscular dystrophy, which is basically the degradation of muscle and the weakening of a muscle, which is quite interesting to me because the cervix is a muscle, and during menstruation or during giving birth, you need contraction there. So the fact that undergoing this testosterone treatment is kind of hindering that contraction is quite interesting to me. Also, it has oncogenic properties, which means it's tumor inducing, and it also has immunoevasive properties, which means it suppresses your immune system. Next, we have MUC16, which is the second most upregulated gene. So this too is overexpressed in multiple cancers. It causes tumorigenicity or causing cells to create benign and malignant tumors. And it also increases rate of metastasis in several malignant growths. And finally, the third most upregulated gene, REX, it regulates erythroid cell expansion, and erythroid cells are basically red blood cells, and it promotes formation of hemoglobalizing erythroblasts. And it is, something really interesting about it is that in comparison to the organs in the female reproductive tract, it is most expressed in the cervix. So basically, the importance of this study and what this tells us is that based off that cluster 13, there's certain genes that are being expressed that normally or typically shouldn't be there. And as we've seen with the top 3 genes that are most expressed, some of them can be quite detrimental to health and can just be concerning in general. So this information could provide us with insight on how to prevent risk of overall health for anyone undergoing hormonal therapy. And I think to further this research, it would be really interesting to see what genes are expressed in other organs other than the cervix, and see what those effects may have on anyone undergoing that treatment. So yes, I would love to give a big special thank you to the NIH and HuBMAP for my opportunity here, to the O'Neill Lab for getting us those samples, and a special thanks to Dr. Brian Gregory and Rongwu for taking me under their wing, involving me in their research, and just teaching me so much this summer. And yeah, that's about it.
Okay, thank you so much, Tatiana. If anyone has any questions, again, you can drop them in the chat. I don't see anyone right now. But yeah, feel free to drop those in the chat and we'll go on to our next presentation. So Ogechukwu, you are next if you want to share your screen.
Yes. Okay. Okay. Hello, good afternoon. Can you see my screen?
Yes.
Okay. Beginning. Hello, good afternoon. My name is Ogechukwu Etwezim and I worked with the O'Neill Lab, and our presentation is Spatial Transcriptomics, the regional importance in the female reproductive system. So examining gene expression. So there are a lot of modes of examining gene expressions, but we're only going to be focusing on a few today. So we have a targeted method, also known as qPCR, which targets a specific gene in order to see the expression of that particular gene. Then we have untargeted expression, which is also known as unbiased, single-celled RNA sequencing. It detects all of the genes that are expressed in a cell. Some of its limitations include that you would have to use all of the tissue sample, and you would lose some of the information about the tissue's architecture and regionality. So a newer method of examining gene expression is known as spatial transcriptomics. Spatial transcriptomics is a molecular profiling method that allows scientists to measure all gene activity in a tissue sample and map where the activity occurs. So why is location important in the female reproductive system? So this is an image of the female reproductive system. It consists of the ovary, fallopian tube, uterus, endometrium, cervix, and the vagina. So follow along with me as I give you 3 examples as to why location matters. So the first organ we're going to look at is the ovary in regards to the oocyte development. So one of the most important roles of the ovary is that it's the location where the egg or the oocyte develops. Oocytes must undergo a growth and development process within the ovary before they are capable of being ovulated and fertilized by the cell. So there's actually a unique physical distribution of follicles within the ovary, with the vast majority of small resting follicles that are located in the outer portion of the cortex, outer region of the cortex, while the growing and maturing follicles are in the inner portion of the medullary. So we don't know exactly why that happens, why the follicular migration happens. So some questions that we hope to answer with spatial transcriptomics include what drives the migration of the follicles? Is it driven by the oocyte, the follicles, or factors that are secreted by cells in the cortex or medulla? So we would need to analyze the oocytes and the surrounding niches using spatial transcriptomic data to be able to answer those questions. The next organ we'll be looking at is the fallopian tube regarding ectopic pregnancies. So ectopic pregnancies are pregnancies that develop outside of the uterus that leads to the embryo not being able to develop to full-term pregnancies. Ectopic pregnancies are most commonly located in the ampular portion of the tube, and ectopic pregnancies can rupture and can be life-threatening. About 2% of women experience ectopic pregnancies. So here is an image of the female reproductive system. So if an embryo does not implant itself within the uterus and implants outside of the uterus, it's known as an ectopic pregnancy. What we found is that when it implants in the fallopian tube area, most of it, 70% of it implants in the ampullary region. If it doesn't implant there, 12% is in the isthmic, then 11% in the fibril, 3% in the ovarian, and 2 to 3% in the corneal region. So we currently don't know exactly what is driving those ectopic pregnancies to happen. So we're hoping that with more data and more use of spatial transcriptomic data, we will be able to determine the driving force of the genes that lead to those ectopic pregnancies. And the next organ we'll be focusing on is our uterus and early pregnancies. So the uterus is the womb in which the fetus develops. In order for pregnancy to occur, an embryo must attach to the endometrium, which is the innermost layer of the uterus. implantation typically occurs to the posterior portion as opposed to the anterior portion of the uterus. So here is an image of the uterus, and here is the endometrium that consists of the stratum basalis and the stratum functionalis. So usually what we typically see is that implantation occurs in the posterior portion as opposed to the anterior portion. However, we don't know exactly what drives for the embryo to implant in the posterior portion. So we're hoping that with further research using transcriptomic data, we would be able to tell what genes, maybe if it's a gene or anything that drives the implantation in that manner. So some of the challenges that we face with using spatial transcriptomic data is, for one, the cost. It's actually very expensive. It's about $40,000 to $43,000 to use spatial transcriptomic data. Resolution. So right now technology hasn't been perfected to where we can just simply see into like single cell. We can mostly just see into like clusters of cells. And then compatibility with archival tissue. So that's simply just referring to how the tissue is stored. So it can either be stored with fresh frozen or FFPE. FFPE just meaning formalin fixed parafilm embedded. So FFPE is actually not very compatible with spatial transcriptomics, just with the fact that since it's been embedded, we would have a hard time being able to like go into the cell and look into it without being able to like break like the disulfide bonds to be able to see it properly. So I would like to extend a special thanks and acknowledgement to the O'Neill lab, Dr. Kate O'Neill, my PI, Addie Johnston, the research specialist. If it wasn't for them, I don't think I would have been able to get through the presentation. I learned a lot and they taught me just a lot about the female reproductive system. I would also like to thank the NIH, Dr. Zorina Gallus, Dr. Ajay Pillai, and Julie Kim, because they were able to like mentor us in how to properly, do a presentation. And I'd also like to thank HuBMAP Consortium. And if anyone has any questions to me, I'll be glad to answer them. Thank you.
Thank you so much for that awesome presentation. So if anyone has any questions, feel free to drop them in the chat. It looks like we had some questions for Stephanie and Tatiana that Stephanie just posted an answer to. So that's great. Thank you guys for taking care of that. If anyone has any additional questions, yeah, you can put them in the chat and we can get some good discussion going. So we've got Kate, you are next up to present. If you would like to share your presentation.
So it's saying I can't start my share until the other person ends there.
Sorry, ended it. Hold on. Oh, stop here. Stop share.
Thank you. So hello everyone, I'm Kate Da Silva and this is my presentation on 3D printed ovarian molds. So the objective was to create a mold for ovaries to prepare consistent histological slices. The idea is that an ovary can be placed and manipulated to achieve regular size samples for further investigation. So I'll be going over ovarian function and anatomy, Dr. O'Neill's sectioning scheme, the analysis of the tissue, and the 3D print mold itself. This was part of the Pixel Lab, which is a Penn Image Computing and Science Laboratory. My PIs were Dr. Allison Pouch and Dr. Jim G. My co-PI was Dr. Kim Jung-Yu. The graduate student that helped a lot was Olivia Sandvold, and collaborators were Dr. Kate O'Neill and Addie Johnston. So ovarian function, ovaries are the primary reproductive organs with three important functions. They protect the eggs of females born with, they release eggs for potential fertilization, and secrete hormones from the corpus luteum. The ovaries release one egg, sometimes more, each menstrual cycle through ovulation, and each ovary contains follicles, and inside each follicle is a dormant egg. So before puberty, ovaries are just long bundles of tissue, and when they mature, they become to be about the size of an almond, but sizes can vary between females. So typically a female is born with two ovaries, and they contain around 150,000 to 500,000 follicles. And I found it interesting that by sexual maturity, these drop to about 34,000 follicles. So when an egg is stimulated by hormones to maturity, the follicle migrates to the ovary wall, and the egg and follicle grow and mature. And these mature follicles are known as Graafian follicles, which can grow up to 1.2 inches or 30 millimeters in diameter. So the follicle contains a fluid that protects the egg as it grows, and when the egg is released, it bursts, releasing the egg to the nearest fallopian tube. From the fallopian tube, the egg travels to the uterus, and if it's not fertilized, then it's flushed out of the uterus through menstruation, which is a cycle that lasts 28 to 32 days. So the anatomy, the tunica albuginea is a condensed layer of fibrous tissue on the outside of the surface of the ovary. The uteroovarian ligament, or the ovarian ligament, is a fibromuscular band that spans from the uterus into the broad ligament. And the infundibulopelvic ligament, or the IP ligament, also known as the suspensory ligament, is what attaches the extreme lateral part of the fallopian tube to the pelvic sidewall. The mesovarium is what suspends the ovary, and it's also where the cut is made in order to do analysis of ovaries. The cortex is the outermost layer of the ovary, composed of stroma and developing follicles, while the medulla is the middle portion of the ovary, composed of stroma, blood vessels, and nerves. And the hilum is what supplies the blood vessels and nerves into the functional tissue. So Dr. O'Neill's sectioning scheme can be seen to the right. First, you start with the whole ovary. Next, it is cut into medial, central, and lateral poles. And originally, she was thinking about doing between 6 to 9 sections, but she decided she wanted 4 sections per pole for a total of 12 sections. So next, each of these sections so it's very hard to see where the cortex ends and the medulla starts. So each section is placed in formalin, and then quarter slices of the sections are created by taking it laterally and cutting them into quarters in sort of a clockwise motion. Then each piece is put through a process called snap freezing, which is the rapid cooling of the tissue for preservation. Next, it's put through OCT, which is optimal cutting temperature compound, as seen here. And this is to mount very thin slices, about 0.5 millimeters, onto slides. From there, the pathologist stains the slides with H&E to determine the normal morphology of the tissue. Then RNA is extracted from the tissue and sequenced and matched within the region, and the localization of cells that takes place is from 3 to 4 cells per region. So originally, Dr. O'Neill wanted to use the Thomas Sadie Riggs tissue slicer, as seen here, but she found that there was some position difficulties, so instead she opted for freehand cutting, and she explained that it was much like cutting an onion. So other disadvantages to the Thomas Sadie Riggs tissue slicer is that the ovary has difficulty laying flat since it's circular or oval shape. You can't adequately view the sections to be cut. There's also size difficulties because it varies from patient to patient, and position difficulties, as mentioned before. So the alternative was to create 3D printed molds customized to ovary side to aid in consistent cross-sectioning for subsequent analysis. So I created the 3D print mold using Fusion 360, which is a computer automated design program that combines shape modeling with mechanical design and manufacturing for product design and development. Python is what I use to automate the mold, as seen here, and you can view the mold running itself. So then for the mold itself, ABS, which is acrylonitrile butadiene styrene, was used, which is a hard plastic, instead of PLA, which is polyactinide, which is another type of plastic. When we printed the PLA mold, we found that the sections right here would fold in on themselves, so it wasn't very sturdy. For the code itself, open source code was used. So ABS was also chosen because it can withstand temperatures from -10 degrees Celsius to 100 degrees Celsius. And the specifications for the mold itself, it creates 12 uniform sections, and each section here is about 0.5 millimeters for the blade. So an advantage was that the ovary lays flat, The sections can be adequately viewed to cut. It also created uniform cuts, and it was faster slicing. So a disadvantage was that ABS printed parts can't go through autoclave due to the heat, so it has to be hand washed. When the mold was tested, shedding of the material was found with slicing, but this can be mitigated by increasing the size of the blade sections. And there's poor conductivity with the mold itself, so this wasn't a problem with this particular project because they were doing the cuts at room temperature. But if anyone ever wants to take the mold and the ovary and keep them together and have them both frozen, you would need a different material. So to the right, you can see the mold in action, and this is it cutting the ovary. I swear it's not chicken breast. So I'd like to thank my PIs, Dr. Allison Pouch and Jim G. I'd also like to thank the graduate student, Olivia Sandvold, who helped with the animations on this project, and Casey Henson for being there to let me know what looked good and what looked terrible about my project. I'd like to thank FJ for the help with Python coding, and I'd like to thank Dr. O'Neill and Addie for the tissue samples and for the images, and thank you everyone for listening to my presentation, and HuBMAP and the NIH for this opportunity.
Awesome. Thank you so much, Kate. That was a great presentation. If anyone has any questions for Kate, you can put them in the chat. I did not see any come through during your presentation, so yeah, if anyone has any for Kate, just put them in the chat and she can respond to those. And we will go on to our next presentation, which is Casey.
All right, thank you. Let me go share my screen. Can you guys see it?
Yep.
Okay. All right, hello everyone. My name is Casey Henson, and for this summer I worked with the 3D modeling of the uterus with magnetic resonance imaging. I worked with the Penn Image Computing and Science Laboratory, or PIXL. My PIs were Dr. Allison Pouch and Dr. James G. And I also had amazing assistance from the graduate student Olivia Sandvold, and my collaborators were Dr. Kate O'Neill and Dr. Addie Johnston. So the purpose of my project was to first provide a detailed 3D image of the uterus and its parts using manual segmentation. I was then tasked on creating an animation detailing the said segmentation in accordance with Dr. Kate O'Neill's slicing protocol for visual documentation, which will be used for instructional and informational purposes. And the objectives of my presentation will be first to discuss uterine anatomy, structure, and its main functions. I will then discuss the main software applications that I used, which was ITK Snap and ParaView and the usage of 3D segmentation. And finally, I will be presenting the segmentation and slicing animation. So the four main structures of the uterus, as shown here to the right, involves the fundus, the body, the isthmus, and the cervix. It has several main functions. First, it provides structural integrity, such as supporting the bladder, the pelvic bone, and several organs. It also serves as the implantation site of the blastocyst during pregnancy, and it provides protection and support for fetal growth. It is also the main site for menstruation. The uterus is also lined with three main layers, the serosa being the outermost layer, the myometrium being the muscular or the middle layer, and the endometrium being the innermost layer. I will then be discussing the uterus dimensions and its variability. So typically the uterus is about 7 centimeters in length, about 5 centimeters wide, and about 2.5 centimeters in thickness. However, variations are introduced with respect to age and gravidity. Gravidity meaning just the total number of pregnancies the person has had with their lifetime, that it could be successful or not. So typically the length decreases after the age of 40. And with pregnancy, it tends to become more flat and rounded with age. A paper that I read from Vergitz et al. Also discussed the length-width ratio and how it decreases with age, with the mean of it being about 1.618 at the age of 21. They also found that this was correlated with successful pregnancies. So this value tends to be the optimal number, 1.618. And as the person increases with age, this decreases, and also the successful number of pregnancies that they have. So then I will be talking about the first main software that I used for segmentation, which is ITK Snap. So ITK-SNAP was created by Dr. Paul Yushkovich of the Pixel and Dr. Guido Gehrig of the Scientific Computing and Imaging Institute. It is a free, open source, and multi-platform software application used to segment structures in 3D medical images. So for my project, I mainly used ITK-SNAP's manual segmentation feature. However, there are also semi-automatic segmentations available using active contour methods. And the interface looks like this. From the left and right, you can see the different colors showing the different segmentations of the structures. It's a pretty cool looking interface. So this was the main interface that I was working with. This is a T2 weighted MRI of the female reproductive system and showing the segmentation of the different structures of the uterus, again, such as the fundus, the body, the lower uterine segment, and then we have here the cervix as well as the ectocervix in different angles. So for my 3D sectioning scheme, I was first tasked with separating the cervix from the body of the uterus then being bivalved, meaning separated in halves, the anterior and posterior aspect. And then the body is separated into 3 sections, and those 3 sections are further subdivided into 3 more sections lengthwise. Better visualization here. So the left image is showing the uterus being bivalved into halves, again, anterior and posterior aspect, and it being divided into 3 sections and further into 3 more sections lengthwise for a total of 9 sections each for each of the halves. The cervix and the ectocervix are then divided into quadrants, and those quadrants are then divided into 3 slices for a total of 12 sections total. Again, here the cervix on the left and the ectocervix on the right, divided first into quadrants and then into 3 sections for a total of 12 sections. The main program that I used for segmentation and slicing and actually making those slices visible was ParaView. So ParaView is an open source multi-platform data analysis and visualization application that I mainly use for post-processing and further segmentation of the 3D mesh. I used Threshold to separate the cervix from the body of the uterus, and I mainly used Clip and Transform to do those slices and then move them along so you can actually see the separation. And I also used the group datasets function so I could slice each section, such as the body, the fundus, and the lower uterine segment all at once, just for easier task. So now I will be showing the slicing animation. I will first begin with the body of the uterus and then the cervix. So here we are with just the uterus and the cervix attached. So again, the main structures, the fundus, the body, the lower uterine segment, and the cervix. We will first begin by separating the uterus from the cervix. So we're just left with the body. And here it is in the lateral view. We will then proceed to bivalve that into halves, anterior and posterior aspects. Each of the halves will then be divided into 3 sections. And those 3 sections will then be further subdivided into 3 more sections for a total of 9 sections each. And here we have the cervix. The cervix and ectocervix are labeled just for reference. And then each of those will be separated from each other. Here we have the cervix and the ectocervix, cervix being shown at the superior view and the ectocervix being shown at the inferior view, both of which will be divided into quadrants. And each quadrant will then be further subdivided into 3 sections for a total of 12 sections at clock positions. So that finishes the animation. I would like to thank Dr. Allison Pausch and Dr. James G for this amazing opportunity. I learned a lot from uterine anatomy and all the way from using all those complicated softwares to Olivia for helping me with the animation and my presentation, made my job a lot easier. For Kate for being there as an amazing lab partner and also helping me with the animation. I would also like to thank Ajay, Julie, and Zarina for this opportunity and the NIH and the HuBMAP program. Thank you very much. That's the end of my presentation. And if you have any questions, please let me know.
Awesome. Thank you so much, Casey, for that great presentation. As we have been doing, if anyone has questions, just drop them in the chat. I don't see any right now, but yeah, Casey will be here if anyone has questions for him. Thank you so much. And we'll move on to the next presentation, which is Jill. So if we want to swap over the screen share.
Hello, everyone. My name is Angel Gates. I'm currently a senior at Howard University, studying maternal and child health with a minor in chem from Hollywood, Florida. And today I will be discussing single cell mapping of the small bowel and colon and focusing primarily on segmentation because that is what my internship was based on. So for the agenda, I'll have a brief overview over what the actual task was, where the collection sites were. I'll talk about Codex multiplex imaging. I'll briefly cover image analysis and segmentation. I'll go over segmentation processes and the errors that occurred, and then I'll have my acknowledgements. So as a brief overview, I had the opportunity and the pleasure of working with the Nolan Lab at Stanford University. And we were tasked with mapping the complexity of the small bowel and the colon. And between the two organs, we had 8 collection sites in total, and you can see them here. I'm just gonna briefly discuss some of them. So like from beginning to end, you start with the duodenum. Right here, you have the duodenum, the mid-small bowel, and then the terminal ileum. You have the ascending, transverse, descending, and sigmoid colon. For Codex multiplex imaging, this is the software that we used in order to process our data. It fulfilled the need for a multi-marker spatial analysis, and it allowed us to see the spatial proximity of the cells within the specific tissues that we were looking at. The Codex system uses 3 main parts, which is barcodes, antibodies, which are conjugated to those said barcodes, as well as reporters, which allow the fluorescent light to appear once the workflow process is complete. This workflow process is 3 steps: is stain, image, and analyze. And what happens is the tissue sample is stained with the entire antibody panel. And 3 at a time, the reporters are added so the fluorescent signal can be given if that marker is found within that tissue. Once this process is done, the next 3 is given, and then the images are layered on top of each other for analyzation. In order to view these images, so we have different ways. The first one is the composite image, which is basically allows us to view the fluorescent signal. And this data allows us to segment. The segmentation allows us to quantify the proteins that are expressed based on the amount of fluorescence that is there. And because of this, we're able to identify the cell types in order for clustering, as somebody mentioned earlier. And knowing these cell types can help us know the biology and understand it better. We are also able to locate the cell tissue for cell type mapping. And we're also able to see other cell types around this area for spatial analysis. Once the process is complete with the Codex machine, we are presented with images such as these. Here we found 6 of the 44 markers that we wanted to locate in this specific sample, and they are shown in these different colors based on whichever marker they were. So again, just to reiterate, segmentation extracts the protein expression based on the amount of fluorescence in the cell, and this is what I focused on. So for the segmentation process, I would begin with an image such as this, where you can barely make out what it is. For those who don't know, it's cell nuclei, and I use a software called Annotator J coupled with Image J in order to get this picture right here on the right. So what this is showing, once we adjust the brightness and adjust the channels within that, we see in the green here, we have the cell nuclei, and in the blue, we have the cell membrane, and we're able to get a better view of this. So once we start segmenting, we are able to get a better outcome. So what segmentation is, before I go in and edit the segments, we receive a segmentation from a previous system, a computer algorithm that gives us these segments, and they're not always correct. So I will have to go in and edit these, and I will talk about the errors in the next slide, but that was what I was focused on for the duration of my internship. So some of the errors that came up was missing outlines, background noises would be outlined, and also some of the outlines for the cell nuclei will be combined as one. So here we see the missing outlines where they just completely fail to acknowledge the cell nuclei being present We have background noise, acknowledge, which is basically anything that's not important to the process. So it's not either a cell nuclei or a membrane, which I will have to go in and delete. And then the last thing is two cell nuclei being combined as one, and you can see it here and here. And this is just due to the closeness of the proximity of the cells, or the algorithm failed to realize it was two cells. So once we go in and fix an entire image, and these images can take anywhere between 3 hours and 8 hours, we produce images like this. And the purpose of this segmentation process is so that the algorithm can give better segmentations to future data sets. So eventually, we don't have to go in and edit these cells or fix corrections that need to be made. So I'll briefly talk about what I learned. I got a better understanding of the small bowel and the colon anatomy. I was able to understand the basics of Codex multiplex imaging. I had a better understanding of cell segmentation. I learned how to navigate Annotator J and Image J, and I had the opportunity of speaking with a lot of people with career paths and personal testimonies. And for my acknowledgements, I would like to thank HuBMAP, NIH, specifically Zarina, AJ, Julie, for giving us all the presenters and the guidance. I would like to thank the Nolan Lab, Gary Nolan specifically, for allowing me the opportunity to just work in the space and be there. I would like to thank John Hickey for guiding me and helping me with this presentation, because without him, it would have not been done. I would like to thank Gulan for providing me with these segmentations, Shakira for helping me learn my way around the lab. Even though it was virtual, she still gave me that one-on-one experience. And everybody here is the people that I met and gave me personal testimonies about not only this internship, but about my career. And I really appreciate that. And lastly, I would like to thank all of my peers who completed this internship with me, because it was not easy at all. And thank you everybody for listening.
Great, thank you, Angel, for that great presentation. That was super interesting. If anyone has any questions, please drop them in the chat. Otherwise, we will move on to our next presenter. We have FJ next. If you would like to share your slides, you can start your presentation.
Yeah, sure, thank you. Sorry, one sec.
No worries.
Can everyone hear the background noise?
No.
Okay, great. Okay. Well, thank you everyone for coming today. I will be discussing a novel redesigned to metadata collection. So to begin, we have to ask, what is metadata collection? Why is it important? And how is it important to genomics and HuBMAP as a whole? So let's start with collection. What is data provenance? Data provenance is where a piece of data comes from. It's kind of like the historical ledger of data. And how is this relevant to genomics? Well, genomics data is collected after a complex set of protocols, all of which can affect the quality and the authenticity of the data collected. And then metadata describes, explains, and makes it easier to manage information in informative resources. So what kind of metadata are we collecting? We are collecting specifically RNA sequence samples. So kind of like the sample ID, the harvest by, which describes the researcher who collected the sample from the source material, be it a culture dish or tissue. The species constitute the species from which the source material was derived, and then the strain is the strain of the source animal. So in the culmination of data, there are problems that arise. Inputting data can be rather labor intensive, especially because it happens when experiments are already completed, making it the task of recalling pre-recorded data very time consuming. And researchers sometimes dread this obligation because it can be seen as an inconvenience to their work, to the basis of their work. So we, the Kim Lab, seek to remove this feeling by proposing a solution where experimenters, lab techs, and the like are able to capture metadata conveniently and while they perform their experiments. Which leads me to my objective: capturing metadata to establish data provenance for HuBMAP before, during, and after procedure while using a user-friendly and uncomplicated input interface. So in analyzing the previous method of data encapsulation, I saw that in capturing a sample of data that, and that is essentially what we're trying to do, collect attributes of samples. So in collecting samples, many of the attributes were analogous. So the similar samples would be categorized into groupings of similar properties and features, and then the groups themselves would share common features, which could then be categorized themselves into batches. So creating a hierarchical scheme as shown on screen. A batch can have a given set of attributes common to all groups. All samples of a group could then share similar fields, allowing a top-down approach to metadata collection, which would make inputting the data much simpler. To further explain, I've captured some images of my hierarchical approach method. So imagine only having to log onto your device of, let's say, 100 samples. So you log onto your device during lab procedure, you have 100 samples. You create groups of samples and batches of groups. So in the batches, you add a field attribute and instantaneously assign an attribute of 100 samples. Or in groups, you assign, you add a field that assigns 45 of the 100 samples. So this significantly reduces the task of individually filtering through and entering the data manually. It also reduces the risk of inputting errors. So as you can see in my hierarchical setup, we have three banks, in a sense, three banks. We have the batches, the groups, and samples. And so we're able to create a batch, and for a given batch, we're able to create as many groups as we desire. And for a given group, we are then able to create a given number of samples. As shown here, we can create the batch. This is the batch view. Then we have the groups where we can see the individual samples in the groups, and then we can see the samples and the nicknames. And from there, we're able to create different batches, different groups, and different samples. To continue in the data entry view, each level on the approach will have its own level of attributes, which will affect the scope of the given level. Meaning in each level will have a species that can be edited within the selected vision parameters. So as we can see, we can look for a certain batch, and with the given batch, we will then be able to input different data. So the same is for the strain, where these attributes also have built-in logic. So depending on what you pick or your selection, different options are presented. So we have here a strain picker for the strain, where we can see the different types of strains that we can select from. And depending on the option that you choose, you can have different images show up. So now to achieve these views to implement this logic, I had to learn Swift UI and Xcode, which is a programming language and a simulation environment, respectively, that allowed me to create these apps, which make metadata collection mobile. In addition, I had to become familiar with IIS core data, which is an object graph and persistence framework. Essentially, it allowed me to persist and save the data upon entry locally to the device. So with the knowledge of core data and the Hub of God family and the Chem Lab, I was able to make a functioning design that I plan to persist in the future. And with that, I would like to acknowledge the NHGRI for their sponsorship, the NIH, specifically Zarina, Ajay, and Julie, for everything they've really done for us as interns. We really appreciate their mentorship and their time and willingness to help us through everything. I would also like to acknowledge and thank Dr. Kim, Dr. Fisher, and Dr. Nordgren for just their support and guidance and the app designs to kind of working through with me. And I would like to thank the Chem Lab as a whole for their support and guidance as well. Thank you, everyone.
Great, thank you so much, FJ, for that great presentation. If anyone has any questions, put them in the chat, please. And we should have time for some additional questions if anyone has them. So definitely start thinking about that if you have questions for any of our presenters, since we just have one more. So Roselkis, you are our last presenter. If you'd like to get your slides up on the screen. And we can get started with your presentation.
Yeah, I think like FJ have to stop sharing so I can share mine.
Oh, I'm sorry. One second.
That's fine. Okay, let me try now. Can you see my screen?
Yes.
Okay. So hi everyone, my name is Roselkis Morla, and I was a summer intern with the Hellenborg lab at Harvard Medical School. And today I'm gonna talk about visualizations of HuBMAP diversity. So the motivation that we have for this was to serve as a reference atlas for future studies in human disease. HuBMAP needs to include a diverse population of donors representing a broad spectrum of factors affecting health. This include age, sex, race, ethnicity, and many others. So this means that in order to have like a diverse population, we need to have donors from different backgrounds so we can have a more diverse population in our database. So understanding HuBMAP diversity. Currently, there is not an easy way to get an overview of the characteristics of all HuBMAP donors. For example, if you go to the HuBMAP portal, you can kind of, if you click on any donors, you can kind of see something like this table, which it shows the characteristics of each donor. So you can kind of see the age, the blood type, the body mass index. But what about if you want to see how many donors are blood type O or how many donors are blood type AB? There is no an easy way to get access to it where you can see all the donors combined. Right now, you can just see one donor, like everything per one donor. So this is where my project comes in place. So I have to create a new page in the HuBMAP portal that contains visualizations that shows the diversity of the HuBMAP data. So the process of creating the page. First, I have to get familiar with the HuBMAP portal UI, and then I have to set up the development environment for the HuBMAP portal UI. Basically, I was able to get familiar with the HuBMAP portal, and then I was able to run the portal UI on my local computer. Then I was able to create a page where I will use to implement the visualizations. I was able to learn about React, GitHub, Elasticsearch, and Chart.js. Then in order to know what type of visualizations we needed, we need to conduct a survey, which I will talk about later. And then I sketch the graphs and tables, and then I implement the visualizations. So this one is a user survey. We implement a survey with 2 questions aimed at understanding which donor properties are considered most relevant to access diversity. I created a Google form for the survey. I share it with the HuBMAP members, Harvard Medical School staff and trainees, and as well as my fellow interns. I received a total of 18 responses, and the results of the survey were used to select the most important combination of donor characteristics for the visualizations. There were 4 characteristics that were really popular, which was age, sex, race, and blood type. So in order to access the information in the metadata, we need to use Elasticsearch. So Elasticsearch is a full text search and analytics engine that allows the store, search, and analyze of a big volume of data. And all the data is stored in the HuBMAP portal UI that contains all the donor characteristics. It can only be accessed through Elasticsearch. So in the right, you can kind of see an Elasticsearch query that allowed me to access to the blood type and sex. So with this type of query, I was able to create tables and graphs. So with the Elasticsearch, with the Elasticsearch query that you just said, so I was able to create this table. So in this table, you can kind of see the distribution of the blood type in the donors, with all the donors divided by the male and female. And this one was the blood type and race. So you can kind of see the distribution of all the donors between like the races. So if you kind of see like how many like wide people are in the donors database have blood type A, you can kind of see that is 6, and you can kind of see which one is the most popular blood type common with the donors, which is blood type O. So this one was the my first sketches with the bar charts. So because I already created tables, we want to create some more visualizations that are more like common and more appeal to the eye. So we created these sketches so I can show my idea of how the bar chart should look like. So this one is a blood type, and in here you can kind of see like there's A donors that are A blood type, and this one shows that there is 12 donors that are O blood type. And this one has two variables, which is blood type and sex. So you can kind of see the distribution between sex and the blood type in there. And then after the sketches and after we got the responses from the survey, the only thing was how we're gonna render all my sketches to the actual page. So I chose Chart.js. Chart.js is a JavaScript library that allows us to render this type of charts. So as you can see, this one is one of examples from the diversity page in the HuBMAP. So this one is sex and race. So with this one, you can kind of see the distribution between the ethnicity and the sex. So if you hover over in the graph, you can kind of tell you like female, we have 22 white female, we have 2 Black or African American female, and we don't have any female that is Hispanic. So what I learned GitHub, React, Elasticsearch, Chart.js, and I also learned how the lab works remotely. Even though it was remotely, I kind of learned how each person contributes to the lab, and it was really interesting to me. And what we learned after creating the page and future improvements is like donors, the donor diversity in HuBMAP can be improved. For example, as you can see in the charts, there is not a lot of like diversity in the ethnicities. So for example, we can have Asians, we can have like more Hispanic. So we can improve that in the future. Some donor might be missing characteristics in the database, and the new visualization page can help to keep HuBMAP and the public informed about progress in recruiting more diverse donors. So for example, with this page, people can see how many donors with different type of races we already have. So they will know exactly what type of donors they will need in the future. So this is kind of like a screenshot of how the page look. I will share the link in a second. And I just wanna say thank you to Dr. Niels, Chuck, John, Tiffany, and also Serena, Ajay, Julie, and HuBMAP and the NIH for funding this. And I will share the link so you can go and see it. So that's the link for the page. Thank you. If you have any questions, feel free to let me know. Oh, is this Serena? Oh, what was the most difficult in building this page? The most difficult part was actually to make the tables in the graph connected to the metadata. So in the future, when they get more donors, all the tables and the graphs will change by itself. So they are really connected to it. That was the difficult part.
Awesome. Thank you so much, Rosalkis, for that presentation and answering that question. If anyone else has questions for her or for any of our presenters, you can feel free to put those in the chat now. We have a little bit of time. So if anyone has additional questions, we still have our interns here to answer them. But yeah, as everyone is saying in the chat, great job to all of our presenters today. You guys had some really awesome presentations and did a really nice job. So great job to everyone. And thank you so much for your time.
And don't forget to send us your feedback on how can we do this better. And I want you to know that you all did fantastic, but we are now counting on you to become our ambassadors. So bring more fantastic interns and also spread the words about HuBMAP in your universities, research communities, or even your communities. As Rosalkis told us, we are very interested in diversity, and if you can come up with any ways by which we can increase the diversity, not only of people who participate in HuBMAP as researchers or trainees, but also donors, we absolutely welcome that. So otherwise, congratulations, everyone. Graduation time.
Okay. All right. Unless there are any other questions, I think we can wrap this up a little bit early, but thank you so much to all of our interns. And thank you to everyone who hosted the interns. And obviously we got some great results, great presentation. So we really appreciate everyone who worked with them and you guys for putting together those awesome presentations. So I think we will wrap this up on this Monday afternoon. And we'll see you all soon.